The title of this presentation is Molecular Biomarkers for Target Therapy in Non-Small Cell Lung Cancer. Lung cancer, according to his histology, can be classified into small cell lung cancer and non-small cell lung cancer. Non-small lung cancer can further divide it to adenocarcinoma, squamous cell carcinoma, and large cell lung carcinoma. Molecular biology of non-small cell lung cancer. Personalized oncology aims to precisely match patients to treatments with the highest potential of success. Extensive research is done to introduce biomarkers which can predict the effectiveness of a specific target therapeutic approach. The EGFR signaling pathway includes several sufficient targets for the treatment of human cancers, including non-small cell lung cancer. Lung adenocarcinoma may harbor both activating and resistant mutations of the EGFR gene, as well as mutations of KRAS and BRAF oncogenes. Less frequent but targeted genetic alterations include ELK, ROS1, and RET gene rearrangements, as well as alteration of MET proto-oncogene, such as mutations and amplification. Except of target therapy, another field is immunotherapy. Accumulation of mutations generally trigger tumor-specific immune defense, but cancer cells have the ability to evade this immune protection by overexpressing some specific proteins. The blockade of immune checkpoint proteins results in potential reactivation of tumor cell killing and induces significant tumor regression in various tumor types such as lung carcinoma. Therapeutic response to AD program death 1 or program death ligand 1 treatment may correlate with the expressions of PD-L1 by tumor cells. In this figure, we can see the frequencies of alterations from the most important genes in non-small cell lung cancer. Genes are called drivers when their genetic alterations, such as point mutations or chromosomal rearrangements, when these alterations are correlated with the pathogenesis and growth of the carcinomas. In non-small cell lung cancer, and especially in adenocarcinoma ones, the most important driver genes are EGFR epidermal growth factor receptor and ELK, anaplastic lymphoma kinases. Secondly, other driver genes are KRAS and BRAF and ROS1. In order to find these driver mutations, we perform molecular diagnostic techniques. These are single gene assays such as polymerase chain reaction and sequencing, fluorescence in situ hybridization, and immunohistochemistry, as well as multiplex gene panel assays using next-generation sequencing on DNA or RNA level. FISH and immunohistochemistry are morphological molecular methods. We can see the alterations of DNA or RNA, or proteins at cellular or tissue level with the aid of microscopes. By FISH, we can detect amplifications, deletions, and gene rearrangements. By immunohistochemistry, we can detect the expression or the loss of specific proteins. Polymerase chain reaction is a method that we can detect mutations or small fusions or amplifications in DNA or RNA after we have isolated genomic material from the tissue block. After this step, we proceed to amplification, and at the end, we proceed to sequencing in order to find the corresponding alterations. PCR is a single gene assay, while NGS is based on PCR methodology, but we can detect mutations, fusions, amplifications, deletions for a large number of genes, for multiple genes, simultaneously, and at the end of the methodology, we need bioinformatics in order to analyze the obtained data and to see the genetic profile of every patient. EGFR mutations were first identified in 2004 and have been associated with response to therapy. Mutations were demonstrated to affect the kinase domain of the receptor protein. This is transmembrane protein. Kinase domain is in the intracellular, and these mutations lead to constitutive activation and downstream signaling in the absence of the receptor ligand. This is the pathway. Two pathways here, or three, we can see, after the activation of EGFR receptor, RAS, RAF, MEK-ERG, PI3 PACT, and JAK-STAT. EGFR mutations were most frequently observed in women and never smokers and show ethnic differences. They are prevalent in Asians. They are more prevalent in Asians than Caucasian populations. The two most common mutations of EGFR, the so-called classical EGFR mutations, are mutation on codon 858 at exon 21, replacing leucine amino acid with arginine amino acid, and the deletion in exon 19 some bases which lead to the removal of 5 amino acids in the corresponding proteins. These two mutations account for 80% to 90% of all EGFR mutations. About 600 are rare mutations concerning exon 18, exon 19, and exon 20. Exon 20 insertions are the most common among them. In this slide, we can see the result of sequencing of 2 patients with lung cancer. EGFR genotyping is generally based on sequence analysis of tumor DNA isolated from biopsy samples. Figure A corresponds to a patient that has the mutation AL858R. We can see in this position 2 peaks. The normal is 1 peak for 1 base. The 2 peaks the 2 peaks correspond to that a proportion of thymine base has been replaced, substitute, by guanine based, and this results to amino acid change leucine to arginine. and leads to a new mutating protein. Patient B has this mutation, T590M, meaning that in this position we can see two peaks instead of one, that is the normal, two peaks where cytokine has been replaced by thymine. And this change at the base and the sequential and the nucleic acid level leads to threonine T to methionine substitution at the protein level. This specific mutation has been correlated to drug resistance with first inhibitors of EGFR. So they have been developed second and third generation inhibitors. KRAS mutations affect about 80% affect codon 12 within exon 2 of the gene. The most common change of codon 12 is the transversion of guanine to thymine, which results to glycine to cysteine or glycine to valine at the protein level. The prevalence of KRAS mutations is 40% in heavy smokers, 30% in former current non-smokers, and 80% in never smokers. This is the Sankey sequencing for a patient with mutation G12C, where we can see here two peaks. The guanine base has been replaced by thymine. This is a point mutation event. And the recent year, this mutation can be targeted with specific inhibitor. BRAF is one of the most frequently mutated genes in human tumors. Mutations are more frequently detected in melanoma, 40% to 50% of cases of melanoma, but are also common in papillary thyroid cancer, colorectal cancer, and non-small cell lung cancer. BRAF mutations are found in 1 to 30% of patients with non-small cell lung cancer, more often in adenocarcinomas and in former or current smokers. BRAF mutations most commonly affect codon 600 in exon 15 of the gene, resulting in exchange of amino acid valine to glutamate of the protein. Here we can see wild-type codon 600, and here this is the mutant codon. In the wild type, the sequence is guanine, thymine, guanine. Point mutation, thymine has been replaced by adenine, and which results to valine replacement, to valine substitution with glutamate. On the right side, these are Sankey analyses for two patients. Patient A has a normal BRAF gene. At this codon is the normal GTG, one peak. Figure patient B has mutant BRAF and especially this specific mutation V600E. We can see two peaks. There are some cells have thymine and some cells have adenine. So the result protein with this mutation is a mutated one and is targeted with a specific protein inhibitor. EGFR mutations, KRAS and BRAF, are point mutations concerning small region of changes in small region bases or nucleacids. Another category of mutations are chromosomal structural rearrangements, and they are involved a larger region of bases at gene level or at chromosomal level. Rearrangements are the result of after a break has been occurred at genes or chromosomal regions, and this break event leads to translocation of bases, nucleic acid, to a small a small part of chromosomal to another region of the same chromosome of another chromosome. These translocations leads after to new genes, new fusion genes, that they are transcribed to a chimeric mRNA, and at the end they are expressed, they are translated to fusion protein, to new fusion proteins. The most important genes that are rearranged in non-small cell lung cancer are ELK, ROS1 and RET, and less frequently NTRK gene. These fusions occur in patient population clinically distinct from classical non-small cell lung cancer, mainly in younger patients with adenocarcinoma histological type. In 2007, the fusion gene EML4-ELK was detected in non-small cell lung cancer. This gene is the result of rearrangement in the chromosomal region 2p2123. This is where the ELK gene is located. And the result is a new protein, a new fusion protein. In this new protein, the extracellular and transmembrane region of ELK are replaced by EML4 regions. Rearrangement occurs in 3% to 6% of all non-small cell lung cancer. It is typically associated with adenocarcinoma morphology, and it is detected mainly in non-smokers or light smokers and in younger ones. We can see the gene rearrangements by the FISH methodology, or we can detect them by NGS procedure. By FISH methodology, we can see at the tissue level if a break event, if it has been occurred. Specifically, with the aid of two different fluorochromes, we label sequences right and left of the breakpoint of the gene, like orange and green. After performing the methodology, we see by microscope the result, and if a rearrangement has been occurred, the two colors are separate. So in this nucleus, we can see a normal gene, the two colors are together, and a gene that has rearranged, and the two colors, green and orange, are separate, giving this specific pattern that is called break apart or split signal. FISH methodology is applied in order to detect rearrangements for all of genes we are interested. So, in this picture, right, these images correspond to fluorescence in situ hybridization, and the left images correspond to immunohistochemistry. By immunohistochemistry, we can detect the new chimeric fusion protein, ELK. Figure A and Figure B correspond to a patient that has ALK rearrangement, where the two colors are separated, and this event leads to overexpression of the new protein, corresponding protein. And this patient is going to receive the specific target therapy. Figure 1C and 1D correspond to a patient that is negative for ALK rearrangement. The two colors are together, and here there is no expression of this protein. Above, we can understand that ALK is present by the brown color in the cytoplasm of tumor cells. FISH method is the gold standard method for the detections of rearrangements for all genes, amplifications, deletions, at tissue level. It's a morphological molecular technique. It requires specific material probes, fluorescence microscope, and experienced molecular staff. On the other hand, immunohistochemistry is more easier technique, wide available, and has a rapid turnaround time. Only for ELK, current recommendation accept the use of immunohistochemistry without FISH in order to test if there is this genetic event, the genetic alteration, because a specific validate antibody and a specific validate platform and automate immunohistochemistry has been developed. ROS1 rearrangements have been detected at first at 2007 in lung cancer. Gene rearrangements are observed in 1% to 2% of non-small cell lung cancer, and they are typically occur in lung adenocarcinomas, rarely adenosquamous carcinoma, and they affect younger patients, non-smokers, and no-smokers. They are often detected at advanced stages, and the patients have frequently brain metastasis. The ALK, on the left, we can see the results by FISH at DNA level, and right, we can see the protein expression. Except of target therapy, where we treat the patients with specific drugs that target the specific genetic alterations, like point mutations and rearrangements. Another field of modern therapy is immunotherapy. Cancer cells have the ability to express certain proteins in order to escape the typical T-cell-driven immune response that would otherwise control them. Immune checkpoint inhibitors work by blocking these specific proteins interactions. Such protein is programmed death ligand 1. This protein is a transmembrane protein and can be expressed both by lymphocytes and tumor cells. In advanced non-small cell lung cancer patients, overexpression of PD-L1 is predictive of clinical benefit with specific inhibitor drugs. The method we use to detect this protein is immunohistochemistry. The protein of programmed death ligand 1 is scored based on the percentage of tumor cells showing partial or complete membrane staining of any intensity. Here we can see that all tumor cells have expressed this protein. They have all their membrane colored with brown color. Immune checkpoint inhibitors are recommended as first-line treatments for stage 4 non-small cell lung cancers without oncogenic drivers and advanced stage of small cell lung cancer, either alone if this protein is expressed above 50% of cancer cells, or in combination with chemotherapy if the protein is expressed under 15% of cancer cells, and this case is negative for PD-L1. In conclusion, in daily routine in our molecular pathology laboratory, we test samples from patients with lung adenocarcinoma for the following markers: EGFR mutation, ELK rearrangement, ROS1 rearrangement, BRAF V600E, V600E mutation, and KRAS G12C mutation, as well as PD-L1 expression by immunohistochemistry. Here we use molecular techniques, and here immunohistochemistry. We test these markers in non-squamous cell carcinomas, and in squamous cell carcinomas, if the patient is under 50 years old and is never or light smoker. This is the current guidelines. Alternatively, we use next generation platforms to investigate these genes, except from PD-L1, simultaneously together with novel investigational biomarkers used in clinical trials, such as MET, NTRK, RET, HER2, NRG1, STK11. Here, in this slide, we can see that during the last 2 decades, the most important genes that have been observed and identified in non-small cell lung cancer and the corresponding target therapy which has been developed in order to target the specific genetic alterations. Thank you for your attention.